Mr. Chairman, ladies and gentlemen, honorable guests, it's a pleasure to be here. Thank you, WIOMSA, for inviting me to hold this keynote presentation. My name is C Öhman in Swedish. People tend to believe I come from the Middle East. They say Dr. Oman, but I can assure you I'm from Sweden, from Stockholm University. And my main interest is marine ecology, fish ecology, and especially the ecology of communities. Recently I went to a wedding and I was sitting beside this lady and she asked me, you know, we Swedes, we like to talk about work. We're pretty boring in that sense. We like to talk about what we're doing, working and so forth. So one of the first questions she had to me, what do you work with? I said, yeah, I'm an ecologist. And she looked at me and she said, wow, I found myself a soulmate. Do you also eat ecological food? And I said yes, and then I didn't say a whole lot more. That's the concept of ecology very much in Sweden, actually. One of my wife's best friends still believe I'm working for the Green Party. Anyway, so I will actually start from a very basic level, from a high school level. I do apologize for that, since this is a very intelligent audience. But ecology is a scientific study of interactions between organisms and between organisms and the environment. Nothing else, at least not in this talk. And the organisms we are going to talk about are reef fish. And the environment is a coral reef. And I said I'm very interested in communities. And actually, I asked this question in a class of ecologists at the university, what is a community? And they couldn't really give a good answer. Well, this is what it is. It's a group of organisms with more than one species. So we talk about reef fish communities. That is what it is. A number of species within a reef. End of high school level. So for us working with communities, with community ecology, then of course the interest is to find out how are these communities regulated. That's like the main question. And if you look at the ecology of reef fishes and how that has developed as a science, it's quite interesting because you can see it's similar to how science have developed in other disciplines. I don't know if it's something with our cognitive process in our brains, but humans tend to like simple solutions, simple explanations. That also you can see in the history of reef fish ecology. 30 years ago, we said that the main reason that we get the patterns we do on coral reefs is because of competition. It's all regulated by competition. And that is what you would call a single factor hypothesis. You try to explain a lot of patterns with one single factor. The discussion went on, then somebody said, no, there's also predation on a reef. You have these big fishes swimming around, and they, through a top-down way, regulate the fish community. And then the discussion went on, and there were all kinds of various things that have been suggested to regulate reef fish communities or any other community. Then somebody a few years back said, no, it's not just one thing. It's many things. And that is what's called the multifactorial hypothesis. I just want to give you a little bit of this background because we will get into, through this talk, various aspects of this. And the one down to the right, that's going to be the main issue of this talk. That is habitat, and how habitat may regulate fish communities. And a habitat is, as you know, the living place of an organism. And it's characterized both by biotic and abiotic factors. So then habitat structure on a coral reef, that's a physical and biological nature of the substratum that includes the corals, it could be the coral rubble, it could be the algae, and so forth. So if you have the reef, the fish, we're very much interested in these arrows. Okay. When we look at communities, these are some of the variables we look at. Of course, abundances, diversity, feeding categories, but also behavior, physiology, and reproductive output. We are all familiar with coral reefs in this audience. We have all seen these kind of sites flying across the Western Indian Ocean, or most of us have been diving on them, built up by these small polyps that live in symbiosis with this algae, the zooxanthellae. And yesterday I heard a very interesting talk of Visram of the zooxanthellae, and I learned a few new things. And we will get back to this algae. And it's this symbiosis that gives us these reefs. In Sweden we also have coral reefs, actually. Many people don't know that, but they're in about 60, 70, 100 meters depth. They grow very, very, very slow. Out of Norway you will also have these reefs. They're quite large. Same thing there, same species, Lophilia. But here in the tropics, what makes it possible that they grow fast is, of course, this symbiotic interaction. And that also explains why we have so many species on coral reefs in terms of fish, because of this habitat and that it's so variable. You will have various degrees of specialization. Various species will associate in different ways with different corals and so forth. This is a study we conducted in Sri Lanka. This is pretty much common knowledge these days, but for butterfly fishes, it's quite common that you see that there's a correlation between live coral cover and the number of butterfly fish. And for that reason, it was suggested that they could be used as indicator species. However, this is not everywhere like this. Actually, one of my students here in Tanzania, Mohammed Noor, or not here in Tanzania, but in Tanzania, Mohammed Noor, he discovered that in Tanzania he did not see these correlations. And here again, we're back to this multifactorial hypothesis. There are many things that could regulate the reef fish communities as we see them. This is another study that was conducted in Mafia Island Marine Park. My PhD student, Kaisa Garpe, spent about 4 months there. And what we saw was that habitat variables explained up to 92% of the variation in fish numbers, and live coral was the most important factor. So out of all these different factors I talked, apparently habitat structure is a very, very important one, explaining up to 92% of the variation in fish numbers. So if we have all these interaction, which will be the main theme of this talk, what happens if it's all degraded? What happens if the habitat is altered? And then we get into the global warming issue. As all of you know, coral reefs or corals are very sensitive to changes in temperature. If you look at the coral reefs around the globe, you will find them in average temperatures between 18 and 30 degrees. But really the figure is, or the range is much more narrow for most corals because they're adapted to the place where you will find them. So corals in South Africa, for instance, will be more sensitive to 30 degrees than corals in Kenya, for instance. Getting into global warming, we have heard a lot about that during this conference. And as we all know, it's due to these greenhouse gases. One of the biggest worries is carbon dioxide. It has increased by 31% since 1750, methane by 151%, and nitrous oxide by 17%, which gives us this global warming issue. Mind you that the greenhouse effect is there all the time. If we didn't have a greenhouse effect, we will all be in a pretty bad situation. But global warming is due to these greenhouse gases, and the temperatures are increasing. Are there scientific evidence for this? Well, it's a pretty strong case, and just to be sure about this, before I left, I talked with one of the climatologists at my university, and he expressed it like this. Global warming is the most likely reason why we see these increased temperatures over the past 100, 150 years. And if you look at this figure, it is a pretty strong case. The red, larger spots indicate that there's an increase of average temperature during the second half of the last century. And this is from 1880 to year 2000. It's a steady increase. And during this period, during the last century, the average surface temperature increased by 0.6 degrees. This may not sound that much, but it's a very big difference. This is, in fact, the largest increase in temperature for the past 1,000 years. And 1990 was the warmest decade. In 1998, which will be the main theme of this talk, it was the warmest year during this whole period. This is something very interesting. You could all get into this website, climatepredictions.net. What they have done is that they have allowed, people have allowed them to use their computers to calculate how the temperature would change over the next 50 or 100 years or so. By doing that, they have increased their computer capacity much more than the earlier models. And what that showed, this was published in Nature on the 27th of January this year. What that showed was that the globally average surface temperature would increase of between 2 and 11 degrees if carbon dioxide is doubled compared to before the industrialism. 2 to 11 degrees. If this is right, we're running into a major catastrophe. I don't know if I will be around in year 2050, but I mean, this is a steady increase. So what about El Nino? Well, El Nino and global warming, sometimes people tend to mix them up. Global warming is a steady increase of the temperature in the atmosphere due to these greenhouse gases. El Nino doesn't really need to be the same thing. It's a periodic disruption of the ozone atmospheric system, which is things that happen in the Pacific. If this is the normal conditions, we have these currents going to the west along the equator. Then between 3 and 7 years, these current patterns changes. And like I say, it doesn't necessarily need to be the same thing as global warming, but same thing there. There's a lot of reasons to believe that they do interact, and some scientists say that the reason we got this strong El Nino 1998 was because of global warming. It was like the first sign on what we're heading for. Okay, this affects corals, and it affects especially zooxanthellae. And again, I could recommend people who want to know really more about this to talk with Visram, because it was very interesting to know about the details. But usually what we say is because of this increased temperature, the coral animal expels the algae, and the corals turn white because they lose their color because algae disappears or is expelled out of the animal. Coral bleach means that the corals are stressed and then they die. This is a beautiful reef area in northwestern Sri Lanka called the Baie Reef. I think besides me and Olle and maybe, I don't know if Ron is here, there are not that many people that had the opportunity to dive there. But this is what you saw when you were diving there. Wherever you looked, it was 100% live coral cover, or at least almost. 100% live coral. And then, after the bleaching event, this is how it looked. Those of you from Sweden may think I'm cheating here, taking this photo in the Baltic Sea, but this is actually a coral reef in Sri Lanka, the same place as this. A photo taken by my colleague Arjun Rajasurya in Sri Lanka. And it's very obvious that these strong El Ninos and coral bleaching interact. Getting back to the study in Mafia Island, you see these columns here. The black part is living coral, the white part is dead coral, and the gray is algae. Again, this was a study that was conducted soon after the coral bleaching event. And many of you that have been diving on Tortilla Reef before know that there was much more live coral there than the 5-10% or up to 20% that this figure shows. Okay, so how does this affect the fish, the reef fish on these, the fish on these reefs? Well, some fish are very specialized, some are more general. This is a very specialized fish. It's a Coravorus filefish that feeds on corals, lives among corals, and if you see the photo up to the right, you can see how specialized it is to move among these branching corals and feed on the coral polyps. And this interesting study from Japan showed that following the coral bleaching event, it started to grow slower, the sizes of the territories increased, it started to eat more rapidly, the reproductive period became shorter, increased competition, and finally it all disappeared. So here we have some interesting community variables again. It's both behavior, it's both fitness, and it's also numbers affected by the coral bleaching event. Another study conducted in Australia also saw that it had a physiological impact on these on a certain butterfly fish. The feeding from Macropora declined, and they became actually thinner. They didn't have the same amount of fat in the system, but they didn't disappear, but they were not happy. Getting into a study which pretty much will be the main theme of this talk is a study that we conducted at Mafia Island Marine Park. Ulf Lindahl, Chris Shelton, and myself. And it was supported by SIDA SAREC Bilateral Marine Science Program and the former SIDA SAREC East Africa Regional Marine Science Program. And in this study, we used quadrats. And all these quadrats, corals were transplanted, fish were monitored. 6 months later, the corals were bleached, they died, and 6 months after that, the fish were monitored again. And what happened was that soon after, there was actually an increase in the number of fishes on these quadrats, which came as a big surprise for most of us, as the big worry was that what will happen with the coral reefs? Are the fish going to disappear if the corals will die? And how will that affect fishery and various things? And one of the groups that really increased were the herbivores, the algae feeders. Because as the corals died, the algae start growing, and the herbivores became very happy. But not only that there were increases in fish densities, in fish abundances, there was also a change in the whole community. This is a multivariate statistics, multidimensional scaling ordination. We have seen it in many talks during this conference. It's a very powerful tool if you want to study communities, whatever those communities is. It was used in psychology at one stage, but now almost any marine ecologist is using it. What it says is that if you have two letters close to each other, or rather as I say, each letter represents a quadrat, or the fish community in a quadrat. If two letters are close to each other, the fish community in those two quadrats are similar. And this is how it came out. The fish community in 1997 before the coral bleaching event, and in 1998 after the coral bleaching event. Pretty different. But what was very interesting in the study was that out of these 32 plots, there were 4 plots that actually survived, that were not affected by this coral bleaching event. And I can also say that those 4 plots came from another clone than the other ones. And there was one talk about Oscar, I don't remember, what was your last name? Where are you? Henriksson. Yesterday about genes and corals and how that varies in the Western Indian Ocean. And apparently there's a genetic aspect to this. Because what we saw was that the 4 plots survived, and the fish communities on those plots clustered with the situation in 1997. Okay, so in that study, the short-term effect was that fish that eat on algae increased in numbers, and fish that depend on live corals for protection decreased in numbers. And the fish community changed. What was also interesting that came out of this study is that even though the corals are dead, it is a habitat for reef fish because the structure is there. I mean, if you look in a dictionary, the definition of a reef is some kind of a structure on the bottom. As soon as if you have a structure of some kind, you will provide a habitat for a range of fishes, in particular reef fishes. So as long as the corals were standing there, mind you, this was branching corals, the kind of corals that was mostly affected by the coral bleaching event, there was still a lot of fish there. And what we also saw that there was a correlation between structure complexity, that is how high these corals grew, and abundance, as well as diversity. And that got us a little bit worried, because since these were branching corals, we thought that, okay, what will happen in the long term as these corals break apart, since we have this correlation? I'll get back to that. First, some other studies looking at short-term effects. This study was conducted in the Seychelles by Spalding and Jarvis. They saw that coral feeders decreased in numbers. That's also a pattern we saw. And they also saw that fish that depend on live corals for protection decreased in numbers. But there were many species that there were no effect. Same thing with our study. Again, going back to my talk when I talked about community ecology, you can kill the corals, but there will always be species that are not affected. And why is that? Well, because the habitat as such, or the corals, are not the regulating factor. There are other factors that regulate those population or communities. Another short-term study, there are not that many, in Australia, they saw that 3 species decreased, and they saw also that the recruitment was affected. And in Arabian Sea, species richness was affected, and they also saw that fish community changed. Fish that eat algae increased in numbers, but there were also other observations which were not the same as we saw. So these were some of the short-term effect studies. There were actually not. That's pretty much it. It's not that many studies that were conducted soon afterwards. Getting into the long-term effects, well, the concept of long-term may imply something more than 6 years, really, especially if you're talking about the history of coral reefs. It's been for a much longer time than humans. But anyway, what happened now? So we did a follow-up study on this, long-term effects of the 1998 coral bleaching event on reef fish assemblages. It was conducted by Kaisa Garpe and Saleh Yahia. Both of them are PhD students at Stockholm University. Saleh is from Tanzania. Where are you, Saleh? There you are. And Ulf Lindahl and myself. And this was supported by the MASMA WIOMSA. It's a MASMA WIOMSA project, the Wildlife Conservation Society, as well as the SIDA SAREC bilateral marine science program. So we had the living corals. The corals were bleached, they died, and then what happened? They eroded. So how did that affect the fish? Well, this is just to show you how these plots looked like. They were not, of course, put in this manner. That would be called pseudo-replication. But in 1997, you had all these plots. They were all living. In 1998, most of them were dead, except the 4 of those. In year 2000, all of them except those 4 were eroded. And then you see there are 2 missing there as well. So those 4 plots were kind of lagging behind one phase all the time. So how did that affect fish numbers and species numbers? Well, clearly the fish abundance, comparing live, dead, and eroded, went down. But if you look at the fish abundance on those 4 plots, since it's only 4, the statistics is a little bit complicated, but still, they follow the same kind of pattern. So the comparison between 1997 and 1998 in the other plots was the same thing as happened in these 4 plots between 1998 and the year 2004, which is very interesting. Species diversity went down, except in those 4 plots. And looking at functional groups, well, the coral dwellers increased and are almost gone in the eroded one and through time. The herbivores that we remember, they increased first and then are more or less gone, except in those 4 plots that survived. And again, looking at the multiple dimensional scaling ordination, you see that there's a clear separation between the living corals, the dead corals, and the eroded corals. And what you should also notice here, which is quite common in community ecology, when communities, whatever they are disturbed, variability increases. So the plots in the eroded are more variable, they're more different among themselves compared to the live and dead coral quadrats. This is a study by Tim McClanahan, Joseph how do you say it? Maina, and Lida Petsöd in Kenya. They also followed up 3 years after, so it's a semi-long-term study, and they found that the hardcore cover was reduced by 74% in marine parks. Open space was colonized by turf and fleshy and coralline algae. They also detected a decrease in butterfly fishes, many being coralivores, feeding on corals, as well as other species. Interesting enough, they saw that surgeonfish, which is a family where you find a lot of herbivores, algae feeders, they didn't really change in numbers, but there was a 244% increase in biomass. What Tim did in this study was to look at both sanctuaries and non-protected areas. These figures are just from the sanctuaries. Okay, one thing that seems to be coming back in this story is that especially species and fish that are specialized, that either feed on the corals or dwell in the corals, are affected. This study by Philip Munday in Papua New Guinea, he looked at how more specialized gobies were affected, or he compared very specialized with those that are more general, meaning that they could move among various different habitats. There was a 59% decrease in acropora corals, and it was also 59% decrease in coral-dwelling gobies. Pretty strong correlation there. Here there was a habitat specialist that was where you found the largest decline. And that's a little bit of a worry, because some of these species were even more specialized than the species we looked at in Tanzania or some other studies. Because some of these gobies, they're found in small population, they have a limited distribution and very specific requirements. So actually, he suggested that if this coral bleaching events carries on due to El Nino, due to global warming, some species may actually go extinct. And that's not so common that we talk about this when it comes to reef fishes, because reef fishes has this pelagic larva phase. They cover, usually species cover very large areas. But actually, there was one of these species, one of the gobies in the study, that they hadn't really defined yet. It didn't even have a name, a proper name, taxonomic name, that they now can't find again, and which they think is due to the coral bleaching event. Okay, so these are some of the studies that have been carried on, short-term studies, and I talked about our long-term study, which we have conducted in Tanzania. But there's a lot of things going on. I can recommend you to attend the session in this afternoon in, I think it's C. Is it here? Okay, it's here. In which Nick Graham, among others, will talk about longer-term studies conducted in other areas. Because our study is part of this big project looking at various sites in the Indian Ocean, long-term post-bleaching effects on fish in the Indian Ocean. We don't have a sexy acronym yet. But Tim, you were going to think about one. Have you? No? Anyway, it includes all these different areas: Sri Lanka, Maldives, Caicos, Seychelles, Mauritius, Madagascar, Mayotte, Tanzania, and Kenya. And it's organized through the Wildlife Conservation Society and Newcastle University, and it's funded by MASMA and the Leverhulme Trust. And you have all these people involved. So it's going to be very interesting to see on an ocean level how this has affected. In that sense, my talk is preliminary data. I had a talk with Nick, and he told me that some of these patterns we have seen in terms of increase of fish algae and effects of coral feeders and so were pretty much the same in Seychelles as in these other sites. Okay, conclusions. Well, we know that greenhouse gases are increasing. The average temperature is increasing. And if these models are correct, we may run into a situation that in 45 years from now, the average temperature has increased by between 2 and 11 degrees. There are reasons to believe that the El Niño Southern Oscillation is becoming more common. I just talked with Olof Linden before this talk, who told me that we are now seeing a core bleaching event in the eastern part of Sri Lanka. The photos I showed you before was in the western part. So maybe we're running into a new core bleaching event. Well, in the short-term effects we saw, and other studies have shown, that corals will die. And again, it's mainly the branching corals. That's important to know. It's mainly the fast-growing branching corals that were affected, which will explain that some coral reefs are not affected as much. In Tanzania, we saw that Mafia Island was affected, but according to Christopher Mohando at the Institute of Marine Science in Tanzania, Zanzibar was not as affected. 6 years, and it's so important also to notice that 6 years post 1998 bleaching in Mafia Island, there is very little recovery. There's actually very little recovery in this area. And I think it was the same in Seychelles, Nick. But there were also in the Maldives, there's more recovery, according to Tim. So these things varies. If we look at the negative in terms of figures, short-term effects, coral dwellers, coral feeders, and habitat specialists were affected to a large extent. And that was noticed in behavior changes. There were negative physiological effects, like sublethal effects. Fish number decreased, and we may see in some sites, for very specialized species, local extinction. Positive short-term effects was that algae feeders and dead coral dwellers actually increased in numbers, or biomass increased. They became fat and happy. But again, these things are not always as easy as we think. There are always a lot of noise. There are species that are not affected for various reasons. As I said, they may be limited by other things, such as recruitment, for instance. There are not that many recruits, or there are many recruits coming in from the pelagic larva phase. Or there may be surrounding areas which have more massive corals, which are not as affected, and so forth. In terms of the long-term effects, well, it was clearly a big difference between the short-term and the long-term. Fish community changed further. We had a different fish community, and it was also an increased variability. If we looked at the Mafia Island site, abundance decreased, diversity decreased, herbivores, coral dwellers, and coral feeders almost gone. So what will happen in the future? Well, the future of reef fish lies in the reef, very much. Since habitat still seems to be a very important factor. This is taken from an article in Science, with two authors on this study from the Department of Systems Ecology as well. And here they hypothesized what would happen if we have a constant bleaching threshold and there is an increase in global warming, and you will have the El Niños following that pattern. Well, if we have the situation A, then there's a big problem, meaning that all the corals would die pretty much instantly all over the place. But that's of course not going to be a case, because you will have different bleaching thresholds. You'll have different species, different depths, different locations, current patterns, and so forth. And the most realistic is probably C, that the threshold will change due to acclimation and evolution. Was an interesting article which I can recommend by Ove Hög-Guldberg in Marine and Freshwater Research, which explains this in a very good manner. He, however, sees that evolution, if this goes on in this kind of speed, maybe evolution will not, the corals will not have the time to adapt. But still, some degree of adaptation will be there. And we should also remember it's not only about the corals, it's about the algae. And of course they have a quicker way of adapting. So what I'm saying is, if global warming and more frequent El Niños, we will have a change of the coral reef system, and some corals may survive or adapt, and the whole system can change according to some of the things we have seen. I called it long term, but really it's only 6 years. The real long term will be in 30 or 50 years from now. So what can be done? This seems to be, global warming apparently seems to be a problem for coral reefs. Well, we could stop global warming. It's easy. Well, it isn't. We all know about the Kyoto Protocol. I don't remember, was it 5% or how many green, was it 5% in 10 years or something like that? Yeah, I think it's something like that. And most of you will realize that that will really have a very little impact. But still, it's a good effort. We should still continue working with marine protected areas. And what this shows, that what is very, very important is actually to protect the reef structure. Continue research. And I'm also a strong believer in artificial and semi-artificial reefs. Looking at the ongoing research in the Western Indian Ocean that relates to this, listening to the various talks through this conference, apparently there are a lot of various research projects in South Africa, Mozambique, Kenya, Mauritius, and so forth, related to various aspects of the coral bleaching. I mentioned the study of post-bleaching effects on fish in the Indian Ocean. We also have Cordio that looks at coral bleaching and coral degradation in various aspects on how corals are affected, as well as a coral reef targeted research project, the World Bank project. So there's a lot of things going on in this region, because there's these things are not 100% straightforward, clear what is going to happen. So we definitely need more knowledge. Finally, I want to say, as I said, I'm a strong believer in artificial reefs. I think that's something we could exploit much more in the Western Indian Ocean. All of you that have been diving on shipwrecks or old Japanese bomb planes, that's the one up to the left, know that it attracts fish. When we talk about artificial reefs, we all tend to get into this discussion, is it really increasing fish, it's increasing productivity and so forth. Well, what you see here is tens or hundreds of thousands of fish in the cold, brackish water of Sweden around an offshore wind power plant taken by one of my PhD students. And I think if it can work in that kind of environment, it should work in the tropics. This is nothing new for you. We all know about the artificial reef. But I think that's something that we can exploit much more, and I think it can be one of many ways of working with this if we're getting into these issues of corals dying and so forth. Okay, thank you.
Thank you, Marcus, for a most interesting presentation. We can have 2 or 3 questions now. Yes, Melita.
Thank you. I'm Melita Samoilas from IUCN. Thanks, Marcus, for a very nice talk. I wondered if you could just let us know what depth you were working in at Mafia. And the reason I ask that is perhaps comment a little bit on the impacts of bleaching in relation to depth. We've been working down in Grand Baie. The work's been done by David Obura, and found some interesting things there in terms of the impacts of bleaching. The reefs are quite deep. They go down to about 35 meters, and anything below 12 meters we were seeing very little impact. So a lot of live coral cover, up to about 70% down at 30 meters. So I'm curious about Mafia.
Yeah, and that's a very good point, because like I said, there are certain corals at certain depths that were mainly affected, and these were quite shallow, it's between 3, 4, 5 meters. So it is in pretty shallow waters. Again, different coral reefs are affected differently, and depth is one of the reasons, and current patterns are another, and so forth. Craig.
Yeah, Dr. Wagner.
Yes, Marcus, that was a good talk. I think the option of artificial reefs it can help, but that will only help the fish, it won't help the corals much. What do you think about the option of propagating resistant clones?
Well, thank you, Greg. I think that actually artificial reefs can help the corals as well, because you will offer a substratum for corals to settle at. But to find clones that are more resistant and to work on that is definitely an interesting one. And this study, this kind of experiment looking at the coral bleaching effect on fish, I mean, as you all understand, we didn't really determine that the coral bleaching event was going to happen. We were just lucky to have the fish numbers there, and that was part of actual coral transplant study. So I think artificial reefs, meaning that you have artificial structures you put on the bottom, can be useful. Again, it's nothing new, but I still think there's a lot that can be done. But from that study, it was this PhD student, Ulf Lindahl, working with this, he showed that what he talked about semi-artificial reefs, that was actually taking coral branches and putting them in these quadras, and apparently was very successful. And as we saw that some of these clones were not as affected, I think that could be a good idea, interesting idea.
Yes.
Is there any evidence that scenario number 3, the coral adaptation, is happening, at least in terms of the short-term data so far? Because I would imagine that depending on which one of the 3 scenarios comes to pass, would have a profound effect on the carbonate's sequestration capacity of your reef systems worldwide.
Yeah, thank you. That's an interesting question. Well, like I said, those 4 plots we saw a change. I don't know if there are any more examples. Maybe somebody else have an answer to that. If you have you seen anything in Kenya, Tim, in terms? Yeah.
There's evidence for it. There is evidence for acclimation. Water temperatures are rising at about 0.1 degrees centigrade per 10 years right now, and that's being used as sort of a acclimation rate that's reasonable for most corals. And so that acclimation is probably occurring for the last 20 or 30 years. And the problem is that the disturbance events, the ENSO events, are actually rising at a faster rate than the background global rise. And so in my mind, the key question is how can you adapt to the difference between the disturbance events and the background events, and which organisms and which symbiotic associations will actually be able to do that. And luckily, nature is complex, so I tend to believe that there will be something to arise out of that disturbance that will be able to adapt. It will have something to do with the host corals themselves, their aspects of their physiology. It will have something to do with the associations between symbiont and coral and their ability to reorganize. And the data that we see so far suggests that there are a whole variety of strategies and differences among corals and their symbionts that will make some new, there'll be some self-organization that will occur and allow acclimation to occur. So a lot of the claims that are fairly simplistic about coral cover and adaptation, the single lines, will be replaced eventually, in my mind, in the next 10 years by biologists that are working on these issues by a more complex sort of reorganization model. And we are working on that. A number of us are working on that.
Thank you. We have, yes, Dr. Arthur.
Thank you. Yes, thank you, Chairman. My question concerns the emerging issue of increasing ocean acidification resulting from the increase of atmospheric carbon dioxide being dissolved into the ocean. I wonder if you'd care to comment on this issue in terms of reef integrity in the Western Indian Ocean.
Well, to my knowledge, there are some studies in the Caribbean that show that this has an impact. How that will influence the Western Indian Ocean, I'm not sure. But the whole idea is that if we get more carbon dioxide in the atmosphere, the sea becomes more acidic, and that affects the creation of calcium carbonate. But no, I wouldn't really know how that has affected the Western Indian Ocean.
Okay, I think we should end the session now. It's been very interesting. For people who want to discuss this further, there's Dr. Öhman and various others here. You can discuss it further. There are also a couple more presentations this afternoon, which are very, very interesting, as Marcus has said. So we now break for the poster session and for coffee. Thank you very much. Then you can go to lunch at 12:30. Before you leave, I have a housekeeping matter. For those people who are staying at the Marina Hotel, there's a bus from this conference center to the hotel at 1700 hours. And from the hotel to the reception this evening at 1830 hours. And from the reception back to the hotel at 2200 hours. Thank you very much.